Now there is another antibody that's been developed, an antibody drug conjugate for the use in lymphoma. We're talking about polatuzumab. It's been approved with bendamustine and rituximab for refractory or relapsed large cell lymphoma. But there's also data that's being presented at ASH 2019 this year about its in combination with lenalidomide and obinutuzumab. And so Lori, that was pretty good data too, right?
Yes, this is a phase 1b/2 study looking at patients very heavily pretreated with follicular lymphoma, 70 to 80% refractory to most recent line of therapy, all chemoimmunotherapy pretreated. Got a sequential addition of polatuzumab antibody drug conjugate, lenalidomide, and obinutuzumab. High overall response rates in the mid 75%-ish overall response rate, which with a high proportion, 65% achieving a complete response. Toxic in that most patients did have at least one adverse event. The majority of patients required a dose reduction or modification, mostly of the lenalidomide component, but serious adverse events were low. So I think that it would be an important potential option in the future, though patients would have to be carefully selected because there's certainly toxicity.
Yeah. And John, there was data that's previously been presented, but sort of builds on this backbone of looking at Pola with obinutuzumab, but actually was not as good as the previous studies with rituximab. I don't really understand why that would be. Maybe it's just a fluke of the patients. Maybe it's maybe it's really not based on biology. But building on this study that we just discussed, how do you reconcile that?
Yeah, well, from a purely pragmatic kind of research point of view, it almost doesn't make sense in terms of what we know about the mechanism of action and what's being targeted by the obinutuzumab and rituximab, that it should work differently with a Pola backbone. But of course, there are differences in the way those agents actually work inside the cell that may give us clues as to why there could be differences. Maybe those can then be overcome as soon as you add in the lenalidomide, which then augments and changes the microenvironment. So we haven't had much chance to talk very much about the microenvironment here, but we know how important that microenvironment is in lymphoma. A lot of that work's tended to be done in the upfront setting. We know much less about the microenvironment in a previously heavily treated patient. So there's all sorts of things that could be happening with not the tumor cell itself, but how in vivo those agents work and interact with what the milieu of cells are that are around there that may impact on it. So, I think you're right. I think the numbers of patients in those studies were really too small to comment. But if you're using an antibody-based therapy here, the way you are with Pola plus maybe you just need something else in these diseases to get you kind of over that hurdle is the first thing. And the second one would be using a chemotherapy agent, which of course is what in the end Pola is. If you're talking to patients about a chemo-free regimen and then you're adding again a chemotherapeutic agent, but at least here you're talking about adding a targeted chemotherapy agent. So I think people find that more acceptable than going back to using chemo with a chemo-free regimen. So at least you can feel that if you're going to have to use chemotherapy, target it and try to decrease those toxicities.
Excellent point.
I was just going to say, I completely agree with focusing on the microenvironment. And as we're moving more towards immunomodulation and immunotherapy, chemo-free options, even looking at gut microbiome. I mean, that's becoming very important in the field. Can that predict toxicity to PI3 kinase inhibitors? I think that's really an emerging field that's going to be important in follicular lymphoma to help select which of these therapies we should be using.